morning, everybody. Many thanks for having invited me to give this short talk about the problem of interpretation of a randomized control trial. I've been interested in this topic for many years. And in this presentation, sometimes a person will appear, the devil's advocate, we say in Italy. I hope that will be understandable also to other people, to ask naive questions and to make some assumption that we will try and contrast. So the first question is why we doctors, who are very busy always during our working hours, should worry about randomized control trials. Mostly because we have to make therapeutical decisions, and these therapeutical decisions must be based on evidence, the best available evidence. Also, it would be a good idea if we could contribute to produce new evidence, participating in research. But this may happen sometimes. Instead of reading a paper to make therapeutical decisions happens every time. So how can we usually make a therapeutic decision? There are three ways. The first one is called induction. So something should work, and should work because in our experience it worked, or because physiopathological mechanisms suggest that it can work. Well, it's not usually a good idea, because medical literature is full of examples in which a good physiopathological idea was gave a wrong result in a proper clinical trial. But anyway, it's a good idea to start a research, to start thinking to a solution. The second mechanism is called seduction or abdication. So we decide just by faith, because our teachers told us to do that, or because we are facing a very rare disease, a very rare condition, and there is one colleague in our area who knows all about that, and we rely on him or her. Yes, it may work in these cases, but in most cases it doesn't work. The good solution is called deduction, that is, to use scientific evidence. And I would stress the first two words, critical evaluation. This means that you shouldn't simply read the paper. You should critically evaluate the paper and then apply what your criticism has shown to be good. This sentence has been written by an old colleague of us, Leonardo da Vinci from Italy. It is written in ancient Italian. I will translate for you. No human investigation can be called true science if it does not use mathematical demonstrations. In other words, research needs some mathematics, some statistics. I have no time to enter in details on that, but always remember that something that is not analyzed properly from a statistical point of view is not a good idea. So you have probably already seen this slide sometimes. This is the pyramid of evidence, and you see as randomized control trial is an extremely important brick of this pyramid. You already heard about systematic reviews in previous presentations. But of course, if you don't have randomized control trials available, you would use other types of research to answer your question. Now, the devil's advocate starts speaking and says, this is a randomized control trial. It is written in the title. So just look at the conclusion and do what the conclusion say. No, it doesn't work this way, my friend. There are many details you have to deal with. And I will report 6 questions about these details, coming from a very old and nice book written by David Sackett many years ago. The first question is, have patients been assigned to treatment or control in a real random way? This is crucial, and we will see a bit more in detail this point. Then, all clinically relevant events have been reported and analyzed. I hope so, but if it is not, you should be able, reading the paper, to check this mistake. Now, are the patients in the study reasonably similar to my patients? I am the reader, I have certain kind of patients. Are the patients described in the study similar to my patients or not? If the study has an age limit, say of 60, and I always treat patients with a mean age of 80, can I use these results? Probably not. Have both statistical and clinical significance been considered? What does it mean? It means that we cannot simply rely on the famous p-value. We need some more message, and we'll be back on that. Is the therapeutic maneuver feasible in my practice? Because if it is not, the paper can be extremely interesting, but I would never use that information because I have not the facilities to do that. And then the last one, have all the included patients been accounted for? Or some patients disappeared more or less clearly from the paper? So let's speak a little bit about randomization. The procedure must avoid any possible influence. No one should be able to guess what treatment would receive the next patient. And the best system to avoid any unmasking is the computer system, a randomized central randomization with a computer system. If needed, the system can stratify or minimize. These are systems to obtain two very well balanced groups. But anyway, the real importance is to have a system which ensures that no one would understand what new treatment, the new patient treatment will receive. The procedure should be understandable to you, the reader, and you should be able to verify that everything has been done properly. Now, some of you maybe have heard this critic already, the paracute critic. The David's advocate says, no one will organize a trial to show that if you jump from a plane with a parachute, your outcome is better than if you jump without. Of course. Yes, but suppose you have two parachutes, maybe one made in Europe and one in China, or one with a different mechanism to open and the other with a different one. How would you decide which one is better? In other words, when the difference in outcome is huge, enormous, like in the case of the parachute, you don't need a trial. But in these days, in modern medicine, it happens very rarely that you are looking for a huge difference. You are interested to show that a small but clinically relevant difference is real. And to show that, you need a randomized control trial. The paper should have a question, a primary endpoint, just one. More secondary endpoints, various secondary endpoints are allowed. They are used to generate hypotheses for future research, but the primary endpoint must be one. And the question must be relevant, both to your patients and to you. Beware of surrogate endpoints. What is a surrogate endpoint? Just think to a new drug which can lower blood pressure. Okay, lowering blood pressure looks a good idea, but what about the reduction of strokes? Or think to a new drug which reduces platelet aggregation. Okay, again, it's a good idea to reduce platelet aggregation, but are you sure that this drug will prevent strokes, ischemic strokes? So surrogate endpoints can be initially studied early phases of research, but after you need proper endpoints. Usually, a yes-no endpoint is easier to deal with and easier to understand for us readers. Dead, alive, relapse, no relapse, recover, no recover. However, in these days, in stroke medicine, there are also other ways to analyze the outcome, like the ordinal shift analysis, also sliding dichotomy. I have no time to enter in details on that. You can find good papers explaining the ways to analyze in this way outcome. But I just mentioned it because you will find in papers on stroke treatment these methods and should be able to understand if they are properly used. Now, a definition of the outcome measures. What is relevant to the patient, what is not. The first step is certainly to define that a biological event happened. So, for instance, you can measure the number of lesions in magnetic resonance. Is it really relevant for the patient? Who knows? It's just a first step in the research, it's not the end. Then you can consider the clinical manifestation of the disease. We stroke doctors measure the NIH stroke scale, but are we sure that a change of 1 or 2 or 3 points in the scale is relevant for our patients? This is, again, a first step, or maybe a second step, but on the end of the research. Then there is disability, which can be interpreted as the independence in common daily actions, and Bartle scale is a good way to measure that. But even better is a mixture of handicap and disability, which takes into account the interaction with the society of the patient. And this is usually measured with the Oxford Handicap Scale or ranking scale, if you like. And let alone quality of life, which is rarely used in trials and it's quite complicated. Sorry. So, I will give you a couple of definitions. The first one is internal validity, what we mean for internal validity. This is the official definition, which is quite confusing to me. But to synthesize, internal validity is the integrity of the experimental design. So, the degree to which a study can establish the cause and effect relationship between your treatment and the outcome is the internal validity. And internal validity is a sine qua non of research. If a paper has no internal validity, if a trial has no internal validity, it's not valid. So you can throw it out. And there are well-known rules to ensure internal validity. Some already mentioned, the sample size calculation or randomization, the intention to treat analysis. I will be back on that in a minute. And all doctors in all hospitals agree about the definition of internal validity. So it should be a universal concept. What can undermine internal validity? The selection bias is what happens when you decide who to treat and whom not to treat. And this can be avoided simply by randomization. Then there is the performance bias. We give ancillary treatments to our patients, and if we give a better ancillary treatment to treated patients and a less good ancillary treatment to control patients, we have a bias. This can be overcome by blinding. Although I would say that it is not always necessary, because some treatments carry with them implicitly some ancillary treatments. And in this case, there is no need to blind. The attrition bias. Attrition bias is what happens when you consider patients who didn't comply with the whole treatment of the study. We know that even patients treated with placebo, if they comply, have a better prognosis than patients who do not comply. How can we overcome this problem? Using the intention to treat analysis. each patient, every patient randomized to a treatment should be analyzed at the end of the trial, even if the treatment was not taken properly. After the intention to treat analysis, you may wish to check a per protocol analysis, that is, only rely on patients who actually took the randomized treatment. But this is just a second analysis. The main, first important analysis is based on the intention to treat principle. And the last one is detection bias. That is the way we evaluate outcome may be different in different places by different doctors. This may be overcome by a central follow-up sometimes. And also, I would say that if the person who does the follow-up is blind to the treatment the patients received, even if the patient and then the local doctor know the treatment, the trial is okay. This is called the probe method, that is a check, blind check of the outcome by a different person. And many trials now in Europe mostly use this system. The other concept is external validity. What is external validity? It is the extent to which the results I'm reading about can be generalized to the real world population. And of course, my real world here in Umbria may well be different from the Edinburgh patients or organization, or from Vienna hospital, and so on and so forth. So, as opposed to internal validity, which is universal concept, external validity is something local. And I will show you a couple of examples. IST3 was a trial with high external validity because the entry criteria were large, so could be applied in many different hospitals. The procedure were very simple, so could be applied by many hospitals. No high technology was requested. EPITET was a very good trial with a very high internal validity, but since patients were very selected, the hospitals were very specialized, and there was an advanced imaging system, the external validity was poor. And you can have many examples of that in your own experience. So, the David's advocate says as last sentence, the paper is statistically significant, the result is statistically significant, so it's okay, stop reading it. Do. No, it doesn't work this way, because statistically significant just means that the probability of the observed difference to be true is a good one, nothing more, nothing less. And you can understand easily that a p-value of 0.051, not conventionally statistically significant, is not so different from a p-value of 0.049, which is conventionally statistically significant. So we clinicians need something more. We need clinically significant results. Given the observed difference, which may be true if statistics say that it may be true, how much can we rely in practice on this new treatment? There are various measures for that. Probably the most simple, the simplest to understand is absolute reduction, absolute risk reduction, and number needed to treat. But anyway, this data should be present in the paper. You should be able to understand that. So to conclude, I hope I'm in time. The general rules to evaluate, in our case, a clinical trial are: be sure that internal validity is there. Check the rules for a good study, but pose a question that is, which answer is as generalizable as you can afford. And a perfect trial, remember that, a perfect trial without a bit of external validity would never be used by doctors. But after having read a perfect trial with high internal validity, you may wish to test the treatment in a trial with high external validity, and maybe you will be able to obtain a proper use for your patients in a different setting. Thank you very much for your attention.
One of the things you talked about was attrition bias. And I suppose the other thing that's important about attrition bias, when you read a paper, is whether they followed up all the patients. And I mean, in your experience, how often do you find it clear that all the patients really have been followed up?
Well, it's not always so clear in papers we read. Usually it is reported that X patients were randomized and X -1 or -2 were analyzed, and the 1 or 2 were lost to follow up. But the details are not usually well described. It's not easy, you know, better than I know, to find all the patients you included in the trial. You have to use different systems, telephone, in Italy probably the best, you use post, mail, but any effort should be applied to obtain a complete follow-up.
Because I certainly feel that there are quite a lot of trialists who report a trial and seem to think it's okay to have 5 or 10% of patients lost to follow up. What's your opinion about that?
I disagree. I think that if you have a trial of, say, 2,000 patients and you lose a couple, it's okay. But if you lose 100, it's not okay.
Because presumably then the follow-up could be different in the two treatment groups, and it actually might make quite a difference.
Absolutely.
So I agree. And then coming on to statistics, you talk quite a lot about, as it were, using the statistics to prove that something works, but equally you might have something that is marginally non-significant, but or not hardly significant at all, but you know, the trial was just not big enough to detect that difference. Again, what advice do you have to the people listening about how to try and decide whether the trial is truly neutral, they've proved it doesn't work, versus, well, they've just not given us a clear answer and this treatment might still be clinically valuable?
Well, from the point of view of numbers, I think that one should look at the absolute reduction and the confidence limits to have an idea of the real neutrality of the results. And from the clinical point of view, I think that if the result is interesting, although not statistically significant, you should probably plan either a systematic review of all the available evidence, or, if this has not already been done, a new trial, larger and simpler, to obtain a definite answer to your treatment effect.
Yes, and I think that's a real issue. I mean, you said at the beginning that you know, really large treatment effects in medicine are uncommon, and as you said, so smaller treatment effects are much more likely, much more plausible. So I guess that's one thing, that when planning trials, people need to think big, and that's a common issue, that when people describe trials or report the trials or comment on the report of somebody else's trial, they say, oh, it was negative. And actually quite a lot of the time, it's not negative, it has definitive proved that something doesn't work. They've just not done a big enough trial, and actually there might be something in this treatment. And I think that's a really, it's an important principle. And interestingly, it's in being tested now in the context of COVID. So the World Health Organization and the UK, so the World Health Organization's got a trial running of treatments that might work in COVID, and similarly in the UK, a trial called Recovery. which haven't been proven to work, but they might work, and the trial's just got to be big enough to prove that they do. So I think there's a real interest in, really making sure that we haven't rejected treatments too early. I certainly think that possibly quite a lot of neuroprotective drugs might have been thrown out prematurely because the trials just weren't big enough. So if anybody's got a question, please go ahead and type it into the Q&A box, but otherwise Stefano and I'll go on chatting about trials. So the other thing
it's a matter of power, in the paper the reader should be able to understand what is the power of the trial. In other words, what is the probability to show something if something is there. And sometimes this is not reported and calculation is not easy, but should be done to understand if it's just a matter of the number of participants.
And I suppose that's the trouble when people design trials and say they want to reduce the proportion of people who are either dead or dependent, and they do the power calculations and realize they need 5,000 patients. They suddenly think, oh, we can't do that, it's too difficult. And that's that's an issue. And I think you again mentioned that if you make things really simple, it may be possible to recruit that number of patients. But I suppose the other question is, do you think there are too many small trials? Do you think we should stop doing little trials? What do you think about?
Well, it depends on the aim, because if you just want to check whether a trial is possible, to check whether the drug or the treatment is dangerous or not, highly dangerous or not, or if you just want to check the biological properties of your treatment, it's okay to do a small trial. It's a pilot trial. It's not definitive. It can't show an effect, but it's just used to prepare a larger trial. Of course, if you want to show a treatment effect, you do need a very large trial.
I suppose what I'm worried about is wasting research effort. If people don't think carefully about exactly what trial they want to do, and is it a really important question, and is it really well designed, and are there enough people in the country or around the world who really want to do this trial? I think some of the problem is that people set out and they're over ambitious about what they can achieve, or they don't design the trial very carefully. So a lot of money and effort is wasted on trials that were probably never going to answer the question. Do you have a view on that?
I agree with you. And I think that after perhaps having concluded a pilot trial, all the doctors involved should join and discuss what is the minimum effect we want to show, and we are happy to accept the new treatment if this effect is shown really. And then if the number of patients is too large, probably you should look at the international cooperation.
Okay.
Doing a trial with a very small sample size, knowing that you cannot give a definite answer, is a waste of time and money.
Okay, and so just one last question from me. We don't seem to have any questions from the audience, which is fine. But what would your top tip be for somebody reading a paper? What's your one most important piece of advice to somebody looking at a report of a trial as to?
Just one.
Yeah.
Just one is difficult. Can I do what they did? Because if I can't, I can avoid reading the paper. So,
in other words, could you deliver this treatment in your center?
Yes.
I think that's a very good point, because again, lots of people test things that are never going to be usable in other parts of the world, yes. And I think that's a good lesson to go away with. Could I ever use this treatment, or will my hospital ever be able to afford this treatment?
Yes.
I think that's a good principle to go away with. So it's been an interesting talk. Thank you very much, Stefano.
Thank you.
And I hope that the epidemic in Italy is going to fade away now that you've had such a traumatic time, and we'll wait and see what happens in Austria and the UK. So thank you to Alina. Thank you to everyone joining us. And just to conclude from my side, Alina, would you like to just tell everybody about the availability of this lecture and its recording on the WSA website?
Yes, of course. So the recorded webinar will be available on the WSO homepage and the WSO YouTube channel. So we'll send out an email shot to alert you once that's done. And for updates, you can also follow us on Twitter @worldstroke.org and @worldstroke.ed.